Is my slide visible?
Yes, ma'am.
Okay. We will wait for some time for more people to join and then we can start the session. Okay, good evening everyone. This is Angira and this is the starting of our live interaction of week 2. So as I had discussed in the previous section, we had mainly discussed about assignment 0 and some questions of assignment 1, wherein we like majorly talked about archaea. We also talked about what are the different kind of like morphological characteristics that it has acquired, so which helps it to survive in different conditions. We also got to know different kind of bacteria and how they react in the presence of oxygen. Also we got to know about mutualism and also parasitism, the difference, and that uh, soybean plant has got nodules where bacteria, that is rhizobium, resides, and it actually is in mutualistic relationship with the plant. So this session will be mainly focused on microscopy, as well as we will deal with a bit of learning about protein synthesis and DNA replication, like the basics. So you might have also watched this in the video recordings that you are provided for the course, but this live sessions are to just brush up the concepts so that it can help you in understanding or clearing your doubts that you might have after the videos. Okay. Okay. To start with, the first question that we have is, Anton von Leeuwenhoek invented a simple microscope that was first to report V animalcules, which was also the first time bacteria were seen under the microscope. Now you have to answer which was the correct option. So I would suggest that people actually take part and respond in audio rather than doing it in text, because I have shared my entire screen and in my entire screen the PPT is running. So I might not be able to go back to check the text. So uh, please respond in audio.
Hello, your voice is actually breaking.
Oh, I see. Okay, let's wait for some time. I'm connected via Wi-Fi, so I hope it will just get better because this is our college
Yeah,
Wi-Fi.
now it seems to be fine.
Okay, okay, okay.
Yeah,
There might be some, some problem in the between, but hopefully it will be fine. Yeah,
right, okay, thank you.
yeah, yeah. So if anyone can come forward to answer this question, anybody of you know who was Anton von Leeuwenhoek? Yes?
I think it's both 1 and 2 are true.
Yeah, okay. Right? It's actually both 1 and 2 are true. Since we did not receive much of response from the other audience, so I would like to give a brief about who Anton von Leeuwenhoek was and what he did for the field of microscopy. Okay, so Anton von Leeuwenhoek was born around 1632, and he actually was the first person who invented the practical microscopes. So he used this, basically how he used to make the microscope was by polishing tiny lenses so as to give them good curvature, and these lenses he would stack up one after the another to give a magnification of around 275 x, which was actually the highest known magnification around that time. Also, he first saw bacteria around the year of 1674, and since back then bacteria word or the term did not exist, so he gave the term animalcules to the bacteria. Just one second, I have to let someone in. So this kind of bacteria that he observed, since the bacteria name was not known back then, so Anton von Leeuwenhoek gave the name of the things that he observed under the microscope to be animalcules. So his studies, since they were the basics of microscopy, they actually formed the pillars of development of bacteriology and protozoology, and any other microscopic techniques that we are learning today, the basics is his work. Because he did so much for the field of lenses and microscopy and understanding the bacteria, so he is known as the father of microbiology. Okay, this is an additional information. Anton von Leeuwenhoek is known as the father of microbiology, and therefore since the first statement was Anton von Leeuwenhoek invented simple microscope, yes he did, and he reported V animalcules, that is what he reported the bacteria to be. And the second statement was the first time bacteria was ever seen under the microscope. Yes, so both the statements are true. Okay. Now coming to the next question. Phase contrast microscopy scans the surface of the sample with probe and maps the topography or height of the force. The next option is atomic force microscopy, passes through the light source aperture which is focused by an objective lens, and an image is built up pixel by collecting emitted photons from the fluorophore in the sample. Confocal laser microscopy uses beam of electrons to create an image of the specimen. Electron microscopy uses a beam of electrons and their wave-like attributes to see with very high resolution. Now you have to answer which of the following statements are true.
Fourth option is the correct answer.
Yeah, would you please like to give me your name?
My name is Sneha.
Okay. Why do you think other options are false?
Because the second statement, where it says that the aperture is focused by an objective lens with an image that is built up pixel by pixel, that should be most likely it should be the confocal,
True.
not atomic force microscopy. And the first statement should be related to the atomic force microscopy. I
Yeah.
think the statements are actually jumbled here.
Yes.
So the fourth option, it says that they use a beam of electrons.
True. True.
So I think that should be the right
Yeah,
option.
yeah, yeah. So you have actually guessed it right. The fourth option is the correct option. Now since you answered and you might be knowing while others might not be knowing, so I would like to explain each of the microscopy mentioned in this question in details. Okay. So phase contrast microscopy, how many of you are aware of phase contrast microscopy, or you have done phase contrast microscopy in your lab or in your course? If there is anyone who has done it, please respond. Anyone, everybody might have done bright field microscopy at least, right? Yes, everybody might have done bright field microscopy. Actually, you have to respond, otherwise the class will become very boring, because it will be me who will be talking. So therefore there won't be much interaction, since it is a live interaction session. So if you could just interact, it will make the flow better. Okay, assuming that you most of you might have done bright field microscopy. So what happens in bright field microscopy is there is a light source and a condenser which condenses the light coming from the light source into your specimen, and from the specimen whatever the light comes out is again, you know, like it is sorry, it is focused on your objective lens so that you can observe the specimen. So generally in bright microscopy is, if needed, we generally stain the cells or we fix the cells to observe it under the microscope, right? So there is no much, because of the like the dye, there is color difference within the background and the specimen. But sometimes we require to image live samples and we cannot stain it with a dye, because the live samples, if we stain it with a dye, it dies. Therefore there is a need to develop a contrast while observing such samples, and this is where phase contrast microscopy comes into play. So as you can see in this image, one second, I'll just, yeah, as you can see in this image, these are all cells. So there is a difference in the background, and there are distinct cells are being observed with a darker color as compared to the background. So this gives us an indication that the cells have been present, and the cells are different from that of the background due to a phase contrast. Okay, so basically phase contrast microscopy translates small changes in the phase uh, of the light, and changes its amplitude, which is also referred to as the brightness, and therefore creates an image contrast which helps us to visualize the sample. Now if you ask me what is the path of this phase contrast microscope, so the path of this phase contrast microscope is as follows over here. So as you can see, there is a light or a illumination source. So from the light, from the light rays goes and it hits a place called annular diaphragm. Now if you look closely, annular diaphragm is a disc like structure which blocks out majority of the light, and it allows a hollow cone of light to move to the condenser lens. Now this condenser lens focuses the hollow cone of light to the specimen. Okay. Now this, in the specimen, certain kind of light is diffracted and certain kind of light is reflected. Now depending upon the diffraction or the level of diffraction, there will be a level of phase shift of the light. Now the light from the specimen will be traveling directly towards the objective lens, which will again, you know, focus it to the face plate. Now here it ultimately, you know, combines all the light that are coming which are in different phase. Now if it is in different, if the light, two light are in different phase, it will lead to destructive interference, and if two light are in you know, same phase, it will lead to better interference. Now this uh, destructive interference leads to high contrast, and this contrast is then visualized by our eyes through ocular lens. So this is how the path of the light travels inside a phase contrast microscopy. So therefore the first statement that it was written, that phase contrast microscopy scans the surface of the sample with the probe and maps the topography or height of the force. This is completely a false statement, because over here no force is being mapped, or there is no probe which is mapping the force. Okay, so this statement is completely false, as over here we can see in phase contrast microscopy we are not scanning the surface, but we are using light to visualize a specimen and creating contrast depending on the phase of light which is coming diffracted from the sample. Is there any doubt regarding this?
No, it is clear. You may proceed.
Okay, now coming to the next type of microscopy, that is confocal microscope. Now anybody has imaged in a confocal microscope or a fluorescence microscope, so as to speak? Why and when are confocal microscopes used? Can anybody tell me?
They are used for live cell imaging.
True, but you can fix a cell and also you can image. It's not necessary that you have to image live cell by confocal microscope. There is a very important feature of the confocal microscope. I need to know that.
For seeing the particular genes using fluorescence.
Yeah, the answer is fluorescence. So, as of now, like in normal microscopy or bright field microscopy, we were nowhere talking about fluorescence. Can anybody let me know what is fluorescence? What do you mean by fluorescence? Suppose you are staining a cell with DAPI. So DAPI is a dye which is known to be the nuclear stain. Okay, so it stains the cell blue, or it stains the nucleus of the cell blue. Now if you have to observe this cell under a microscope, then you would obviously prefer a confocal microscope or a fluorescent, like initially you will prefer a fluorescence microscope, but fluorescence microscope, there are disadvantages of having a background. So you can see the cell as well as you can see the background. So in order to avoid that background, confocal microscope has come into a picture, and the confocal microscope is a very efficient and a very good it provides very good resolution image of the object that is being observed under the microscope, and the object should necessarily have a fluorophore which is to be observed. So what happens, like here also there is this laser. Okay, so previously we were talking about light source, here we are talking about a laser. Okay, so this laser has a laser goes through, you know, a pinhole aperture, so it allows only a small beam of light to enter into the confocal area, and then there is this excitation filter which actually uh, puts on some excitation rays, and that is pointed towards the sample. Now this light that is reaching the sample is then deflected back by the dichroic mirror towards the objective, and from there we are observing it on the and from there we are getting the light through a fluorescence barrier which cuts out any other light that is coming from the background, and we are only getting the light rays which are corresponding to our sample of interest, and then the light rays finally goes towards the detector which we can observe, and therefore we get high resolution images. Okay, now confocal microscopes are also of two kinds. So the two types are confocal laser scanning microscopes. So this have got a special kind of mirror which scans linearly through the x and the y axis of the the plane or the specimen. and this forms a Rector scan pattern and gives a very high resolution of the sample that you are imaging. But since it is doing a scan in a Rector scan pattern, that is, it will scan like this. Suppose this is your sample of interest, it will scan from here to here to here to here. Therefore it will give very good resolution for fixed cell images, but it cannot give good resolution for live cell imaging. And that is why when somebody over here said that confocal microscope is essentially used for live cell images, it is not, because laser scanning type of confocal microscope are like not working, or not the best method to image live cells, because if you have to use live cells, if you have to have very high speed detector to detect it. Therefore confocal laser scanning microscope are only used for high resolution images of fixed samples. Now if you have to image live cell images, then you have to go to use spinning disc confocal microscope. Now what is there in this kind of microscope? This microscope has got a special, array of pinholes. Previously we saw only one pinhole. Here there is an array of pinholes on the disc. So this disc starts rotating, and therefore the light coming can be like the immediately the light coming out from the samples at a given time will be immediately coming out of the pinhole, and it will be reaching the detector, thereby allowing us to observe the motility of the cells inside the specimen. So therefore this kind of microscope can allow us to observe live cell images. Okay, so therefore the statement of confocal laser microscopy, which uses a beam of electrons to create an image of the specimen, is also wrong, because over here again laser is used as a excitation source and not electrons. Now as I said over here, the scanning of the confocal microscope and spinning this is completely different. The scanning speed of laser scanning is slow, because it scans in a Rector scan pattern, and therefore it gives like low sensitivity but high resolution. Whereas for spinning disc, the speed of scanning is fast, because there is a pinhole that is completely rotating and the light is constantly coming out from each of the pinholes. So you can detect even slight movement of your sample inside the microscope, and the sensitivity is therefore very high, but the resolution is low as compared to point like the laser confocal microscope. Now coming to electron microscopy. So electron microscopy does not use light as electron source. It uses a beam of accelerated electrons as a source of illumination. So what happens is that there is a and why does it use electron and why not light? Because this kind of electron microscope is used to have very high resolution images, which is not possible for the case of any light microscope. Since the wavelength of the electron is about like some 1,000 times shorter than that of visible photon light, so therefore this electron microscope are used to visualize images in order to have high resolution of the images, or we can observe the images in very high magnification. Now the electron microscopes investigate different kind of stuffs. Most importantly, it is used to image wide range of biological specimens, like individual bioorganelles. Sometimes it can be used to observe nanoparticles which are of the order of few nanometers. It can be used to observe virus or any other smaller organelles, so which cannot be observed in light microscope. Now there are two types of microscopes as compared like in electron scanning microscopy. Like in electron microscopes there are two categories. The first one is transmission electron microscope. So in this microscope there is a beam of electrons which is transmitted through the specimen to form an image. So the specimen that is used has got a very like it is inserted over here in the specimen spot, and it is very thin, and the thickness should be less than 100 nanometer, and when the electron is passed through and through the sample, it creates it creates an interaction of the electrons within the sample, and that is goes back to the detector wherein the image is captured. Now what is the emission source, or what is the uh, light source, or the electron source? The electron source of TEM is actually a tungsten filament or a lanthanum hexaboride single crystal source. So this electron beam is generated by connecting it to a very high voltage source, and it emits electron into the microscope vacuum tube. So it is very important for this microscope to function is that this microscope should be completely operating in vacuum. Now in order to focus this beam towards a specimen, there is use of different electromagnetic lens. This electromagnetic lens focuses the this electron beam towards the specimen. Then the electron lenses are also arranged in such a manner that they help in amplifying the electron beam that is reaching the specimen port. Now once the beam passes through the specimen, this specimen, as I mentioned, is very thin and it should be less than 100 nanometer thickness. It then goes and hits the detector, which is a fluorescent screen, and then the image is formed on the detector. So this image of the specimen are then assorted and given different kind of shades, and it is then observed on the computer which is attached to the microscope. Now as you can see over here, this is a TEM image of a cotton phloem tissue. Now if you are to see over here, you do not observe any colored features, because over here we are only seeing black and white images, and all the organelles over here, this is the nucleus, and these are some chloroplast, as you can see over here. These are the vacuoles. So you can see all the features which are not clearly seen even in the light microscope or confocal microscope, so as to say, can be clearly observed in that of the transmission electron microscope. Now one can obviously ask, how can you prepare the sample, because we say that the sample needs to be very thin. Now our samples might not be that thin, so how do we have to prepare the sample? So the materials which are very thick, they needs to be cut down with the help of like with the help of diamond cutter, so as to produce very thin slices, or it can be an ultra microtome can be used to produce very thin slices of the specimen. And also if you are having a kind of liquid sample, then this liquid sample can be drop casted on this grid. This is called a TM grid, which is inserted inside the sample port of the microscope. So if you have to observe any kind of copper nanoparticle or silver nanoparticle, you have to drop cast your suspension on this TM grid, and this grid is very small, less than 1 centimeter in less than around just a few in dia, around 3 in dia. So you have to drop cast around 5 microliter of the sample onto the the grid, and you have to allow it to dry up for some time. Then you have to observe it under the TM to see if the nanoparticles can be observed. Also it is very important that your sample must be completely dried while observing, because water can actually interfere with the resolution of the image. Now, so as to give an reference of how images look in TEM, you can see this is a section of a Bacillus subtilis cell. This is the cross section of the cell, and you can see that these small hair-like structures are the flagella or the cilia of the bacteria, and you can see there is cell membrane also can be clearly seen, and inside of the cell the cytoplasm is also clearly labeled. So we can see very small details of the cells in high resolution with the help of electron microscopy, because it uses electron as a source of excitation and not light, and electron has got a it has very low wavelength as compared to that of light source. Now coming to the next type of microscopy, that is scanning electron microscopy. Now scanning electron microscopy is also works very similar to the transmission electron microscopy, and it produces images of the sample by scanning the surface of the sample with electrons. Or in transmission electron microscopy, we were passing the electrons through and through the sample, but in scanning electron microscopy, it does not happen that way. There is a sample of electrons which scans through the sample, and once the electron interacts with the sample, it interacts with the atoms and produces various different kinds of signals, and each such signal that is produced has got certain kind of information about the surface topography of the sample. And therefore we can say that the method of scanning this kind of used in scanning electron microscopy or SEM is raster scan pattern, the similar pattern that we have also seen in confocal microscopy or laser confocal microscopy. Now over here you can see that here also there is electron source, and there is condenser lens, which are basically electromagnetic lens that condenses the electrons towards the sample, and after the electron beams hits the sample, it is then focused back towards the objective lens. From here the light or the electron goes towards the detector where the sample image is produced. So over here you can see this is the SEM image of pollen grains. So you can see how distinctly the surface morphology of the pollen grains have been shown, and each of the structural features or the small perturbation on the surface of the pollen grain can also be seen, which is not possible to see in light microscope. So basically all electron microscopy work by enhancing the resolution of the image, which is not possible to achieve in the case of light microscope. Okay, so when we are talking about scanning electron microscopy and how it actually images the sample, then we need to know how it interacts with, or how the electron interacts with the sample, and how it generates a signal. So when an electron beam hits the sample, it results in two types of electrons coming out. One is the backscattered electrons, and another is the secondary electrons. Now if the secondary electrons they are only like few nanometers from the top of the surface, so they can escape and originate from the atoms of the sample, and they result due to the inelastic interactions of the electron beam with the samples, whereas the backscattered electrons are electron beams that are reflected from the samples by elastic scattering, and therefore they have much higher energy as compared to the secondary electrons, and they merge very much deeper into the location of the sample. Now since this backscattered electron information are coming from the deeper regions of the sample, therefore they carry different types of both of this scattered electrons carry different kind of information, and each of this information gives us information about how the topography of the sample, or the topography of the surface of the sample. Now over here also the sample preparation is done so as to fit small amount of sample on the specimen stage. So basically there is a carbon tube or the carbon tape to which you attach the sample, and then you keep it or mount it to the specimen stub. The specimen stub size can vary, ranging from a few millimeter to few centimeters, and then what is done is that your sample is then coated with an ultra thin coating of gold or palladium, so as to like if your sample is non-conducting, then it needs to be coated with gold or palladium, so as to generate a negative copy of the samples with the help of this gold and palladium, and then this helps us in imaging the object better. Now coming to atomic force microscopy. So as someone rightly mentioned, that atomic force microscopy, it observes observes an images by scanning the surface of the specimen and not by falling light on its surface. So as you can see over here, in AFM there are different parts. So the most important part is that of a cantilever. So what does a cantilever do, or what is a cantilever? A cantilever is basically a silicon or a silicon nitride, and this cantilever actually, when it moves through the sample, depending on its topography, it faces deflection, and this deflection can be due to mechanical contact force or van der Waals force or capillary force, and this deflection sensed by the cantilever is sensed by the detector in turn, and any movement of the cantilever is reported, and this movement of the cantilever thereby gives us the change in height of the sample, which again gives us the understanding of how thick or how rough is the sample. Now coming to different types of atomic force microscopy. So atomic force microscopy is known to be of three modes. So it can be of contact mode, that is, in this kind of mode the cantilever or the tip is dragged across the surface of the sample and it physically touches the sample, and any deflection in the cantilever is judged as a feedback signal. In tapping mode, the cantilever is driven or it oscillates up and down near the sample so as to form a frequency or resonance frequency. This initially this cantilever tip is brought close to the sample at a very short range, where it becomes where it can actually detect the forces, and any forces which can cause a any change in the height of the cantilever is detected, and that gives us the information about the specimen. In non-contact atomic force microscope, this is done for cases of where the sample is extremely delicate and any kind of touching or contact can actually rupture the sample. So in this kind of microscopy, the tip of the cantilever does not exactly touch the sample. They oscillate as at a resonant frequency, and they they get to know the van der Waals forces, which acts or which decreases or increases the resonant frequency of the cantilever, and on detecting the van der Waals forces of the with which the sample and the cantilever interacts, they gives a signal or they gives a feedback to the detector, which then plots the deflection of the cantilever versus the distance, and that gives us a topographic image of the sample. So therefore this is actually a briefing of all the different kind of modes and its advantages. So in the contact mode has advantages of having high scan, but the disadvantage is that it can distort the image or it can distort the sample, and it cannot be used for soft samples. For tapping mode, the resolution is higher, but the speed is very much slower as compared to the contact mode. For non-contact mode, it is the no damage to the sample is done, and therefore it can be done for soft sample like cells, but the problem is that I have to let someone in. But the problem is that it is only applicable for extremely hydrophobic samples which has got a minimum fluid layer, because that can interfere with the uh, AFM detection. So basically the AFM is used to distinguish cancer cell and normal cells, because there will be some difference in the hardness of the cells. Also it can evaluate different interactions between the different cells, and also it can show us how the stiffness of the cell has how a particular condition has led to the stiffness of the cell. So thereby AFM helps us to understand if a condition, or it helps us to understand how the stiffness of the cell is changing with depending on the condition where it is being provided. Now coming to the next question. This is dash is a mixture of salts, sugars, amino acid, nucleotides and many other biomolecules. The options are plasmid, cytoplasm, ribosome, DNA.
Cytoplasm.
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 Yes, the answer is cytoplasm, because uh, cytoplasm is basically a gelatinous liquid which is present inside your cell, and it is composed of water, salt and any other inorganic or organic molecules. And if you are to ask me what are the what is a cytoplasm and what are its important function, then cytoplasm actually allows to maintain the cell turbidity and or turgidity, which enables the cell to hold their shape. And this fluid, or this a particular structure which is composed of salts and water, they act as different parts of the cell membrane or the cell organelles. And it also is the home of many kind of different activities, such as enzymes enzymes are present on the cytoplasm, which takes or which degrades the waste in the cell. Also cytoplasm, because of its constituents, it assists in different metabolic activities like providing electron donor or electron acceptor. Also it provides a medium for the cells to for the constituent of the cells to move around. So basically it is a matrix wherein all the organelles are suspended. Now coming to the next question. Translation is synthesis of proteins by ribosome. Topoisomerase helps in unwinding the tightly bound double helix structure of DNA. So you have to answer which of the following options are true.
A and B both are independently true statements.
Okay. What do you mean by independently true statements?
It means that both are correct with respect to their functions that is given here.
But both are not both occur in independent events basically, right?
Yes.
Yeah. So can anybody tell me what is translation, or if anyone can explain what is central dogma? Anybody is aware of central dogma theory?
DNA upon self-replication will produce multiple copies of DNA. And then when it undergoes transcription, it will produce mRNA, which is then further translated into proteins. So this
True.
is the
Yeah.
central
This
dogma.
is a central dogma. Yeah. So basically, what you said is true. Both A and B are independently true statements, and therefore, since over here the first statement is about translation, it is important that we know what is the process of, like, what process of translation is. So the translation is basically, it is a process of protein biosynthesis. It is the process by which mRNA is mRNA codes for a particular protein, and that protein comes from the mRNA which has come from the DNA. So there is a particular chain of amino acid that is formed uh, on the like on the mRNA, depending on the code of the mRNA, and this amino acid chain arranges together to form the primary structure of proteins. So if you are to see over here, this is a ribosome. The red colored structure is a ribosome, and this is a messenger RNA or mRNA, and each three code that you can see over here, which is CGC or AAG or CCU, each of these are individual codes which codes for a certain amino acid. Now if you are to ask me, how can this code for an amino acid? So basically this code is detected by transferryl RNA or tRNA, and this tRNA recognizes this code and it brings in the amino acid that is responsible for this code, and this amino acid is then, you know, bonded with the previous amino acids which has been coded so far. So this is the uh, whole process of protein synthesis. So the initial process starts with ribosome assembling around the position of the target mRNA, and the first tRNA attaches to the ribosome, and then the elongation part starts, wherein the tRNA recognizes the codon, and it brings in the protein of or the amino acid of interest and recognizing the codon, and then when as the translation proceeds, it comes across a stop codon, wherein an instruction is laid that the translation has to end. This is when the ribosomal complex remains intact, but it falls off from the mRNA, thereby leading to the formation of a complete protein molecule. Now, since we talked about topoisomerase in the previous slide, we have to know where does topoisomerase take part. So, since it is talking about uh, double helical structure of DNA, so it is quietly understood that topoisomerase takes part in DNA replication. Now if you have to if I would tell you what is DNA replication, then DNA replication is basically forming uh, like a daughter DNA from a parent DNA. So as you can see over here, in order to form a new daughter strand, the parent strand needs to be opened up, and then it will act as a template for synthesis of a new daughter strand. Right? Now once you open up the DNA, it is very important that you have to have certain enzymes that helps in the process. So the first enzyme that helps in the process is that of DNA, DNA helicase, which actually is the protein responsible for stabilizing the or opening the two strands of DNA so as to form this replication fork of the DNA. The next enzyme that takes part in this process is DNA polymerase. So this particular part of the protein, it takes part, it actually starts synthesizing new or the daughter DNA in 5' to 3' directions, keeping the template of the parent strand. And then comes single strand DNA binding protein. It also plays an important function. It binds to the certain bases in the DNA which has been opened by the DNA helicase and prevents it from reconnecting back with each other. Now comes topoisomerase. So topoisomerase, what it does is when we open a chain of a loop, there is a chance that a supercoiling will take place over here. So in that case, topoisomerase relaxes the DNA from its supercoiled nature, and as a result it prevents the DNA replication from stopping, because if the supercoiling happens to a greater extent, then the DNA helicase cannot move forwards and the replication will be stopped. So it is very important that after time to time topoisomerase removes the supercoiled nature of the DNA, so as the DNA helicase can proceed further. Okay, and then comes DNA ligase, which joins the certain parts of the DNA or of the Okazaki fragment which were not ligated together. It ligates them, thereby forming the complete dotted strand. So basically, if we have to talk about DNA topoisomerase, so the main function of this DNA topoisomerase is that it removes or it prevents the intertwined nature of the DNA double helix, because if it is not prevented, or if the supercoiling of the DNA during the replication formation is not stopped, then there will be no further movement of DNA helicase and DNA replication will be stopped. Now coming to the next question, which is very easy. What is the sequence of 3 nucleotides that correspond to a specific amino acid or stop signal during protein synthesis known as?
Codon.
Yeah, this is very easy, everybody knows. The answer is codon. Now, as I said, each particular mRNA codes like has a particular sequence of this kind of base pairs, and 3 of those pairs together is known as a codon, and depending on how these 3 are arranged, they will code for a specific protein. For example, AUG codes for methionine, which generally is the start codon, and then GUC codes for valine. For serine is coded by AGU, and CCA is coded by proline. So over here you can find a table of which codon codes for which amino acids. So basically, apart from coding amino acid, there are codons which asks or gives signal for the stopping of the translation sequence. So this particular codons, that is UAA or UAG or UGA, are the codons which are responsible for stopping translation. So not only does codon code for a specific amino acid, it also gives stop signal during protein synthesis. Okay. So next question is, which one of the following statements is not true about protein structure? The primary structure of proteins is merely chain of amino acid connected by peptide bonds. The second option is, secondary structure of protein alone gives protein its specificity to the target compounds. Tertiary structure of protein is its 3D conformation and is insensitive to chemistry and temperature. Fourth option is, quaternary structure are biological units of multiple protein domains. So can anybody give me an answer? Actually, I want to know what you understand by primary, secondary, tertiary and quaternary structure of protein. So first you can answer that, and then we can talk about what is the right answer. Yes, anybody can answer.
So primary structure, it is just a simple polypeptide chain,
Yeah,
and secondary structure we have alpha sheets, alpha helices, beta sheets, beta barrels, and all of that.
yeah,
They have their respective phi and psi angles according to the Ramachandran plot. And tertiary structure, it is the 3D conformation, and quaternary is just an assembly of multiple protein domains. So that
right.
is it.
Yeah. So for those who don't know, the correct option is actually, which is not true, is secondary structure of protein alone gives protein its specificity to the target compounds. This is absolutely untrue because secondary structure does not have any active site. So in order to understand the structure of protein, let's start from the very beginning. So the first structure or the first step or first hierarchy of the protein is the primary structure. So inside the primary structure, the protein is organ and like it is formed out of ordering the amino acid, and each of these amino acid is joined by a peptide bond. And therefore there is a, there is importance of how this amino acid are attached, because depending on the sequence of the amino acid, there will be bond formation between this amino acid. This bond can be van der Waals interaction, hydrogen hydrogen bond, and depending on what kind of bonds are forming and at what frequency the bonds are forming, this primary structure will lead to the formation of a secondary structure. Now if you see, this is an alpha helix, which is a secondary structure. So what happens is that the primary structure locally folds into structures which does not no longer exist in simple polypeptide chain, and they fold so as to, interact the amine and the carboxyl group of the peptide link. This kind of structures also help in giving the protein chain better stability. So basically the two types of structures that exist are alpha helix and beta sheet. Okay, so over here, as said that there is specificity to target compounds, no, there cannot be specificity because there are no active site presence. Only amino acids are joined by one another to form either alpha sheets or beta helix. Now coming to the tertiary structure of proteins, um, so in this structure, or this structure basically arises from further folding of the secondary structure to, the bonds formed or the folding happens due to hydrogen bonding or electrostatic bonding or disulfide linkages, or there can be van der Waals interaction between the amino acids. This tertiary structure which is formed, it represents the overall folding of the polypeptide chains, and it gives rise to the major molecular shape, and therefore this tertiary structure can be further grouped into two types. It can be either fibrous or it can be globular. Okay, so now coming to the fourth type, that is quaternary protein structure. So in quaternary protein structure, the arrangement, it is the arrangement of the tertiary structure in a different spatial arrangement. So what happens is that the proteins which are composed of more than two polypeptide chains, they arrange in different spatial arrangement to one another, and they form a quaternary structure. So this quaternary structure, depending on the type of amino acid present on the surface, will have its own unique and biologically active three-dimensional sites, which can be also called active sites, and they come also contain a combination of secondary part, and these portions of this quaternary structure are also, which perform a specific function, are also called domains, and today they are known to be the building blocks of protein. So over here we learned that the protein hierarchy can be dependent on four different types. The first is the primary structure, which can fold onto itself depending on various kinds of bond, the hydrogen bond or peptide bond, and it can fold into alpha helix and beta sheet. And this secondary structure, which is alpha helix or beta sheet, they can fold again among themselves to generate a tertiary structure, which then can again specially rearrange itself to form a quaternary structure, and this quaternary structure has got active sites, which was referred to in the previous question. So with this I end the live interaction for today. So in the next class we will be focusing more on the equations that has been like put in assignment 3. So if anybody has doubt regarding the class that, or any doubt regarding the interaction we have today, can write it or ask me.
No doubts.
Okay, so those who don't have doubts can leave the meeting. We will meet again next Tuesday.
Thank you. Good night.